Hello, my name is Brandon Chatfield. Welcome to Onward Principles of a Powerful Mind, where we discuss science-backed ideas to improve your brain, your mind, and your life. Welcome to Onward podcast. My name is Brandon Chatfield. I am an adjunct professor here at NAU in Arizona, and I'm also a psychotherapist. This podcast is separate from my role at NAU and in my practice as a psychotherapist. I am trying to help educate people on various mental health and physical health topics as a free resource to the public. This is not meant to replace professional diagnosis or suggest any type of treatment. For your mental and physical health needs, it is best for you to consult a mental health professional or a physician. It is very important for the listener to take responsibility for their own health and wellness. Thank you. Welcome to Onward podcast, Principles of a Powerful Mind. My name is Brandon Chatfield. This is going to be episode 3, and it's really about how action potentials work and how the human nervous system communicates with itself. So imagine trying to eavesdrop on a secret conversation happening in your brain. No, it's not a spy thriller. It's the fascinating world of measuring action potential in the human nervous system. Let's dive deep into this adventure, complete with electrical potentials of axons, electrodes, microelectrodes, and all those fancy terms that make neuroscience sound like rocket science. To tune into these electrical conversations, we need special gear, electrodes and microelectrodes. Think of electrodes as the big, clunky headphones your dad wore in the 80s. They're good for picking up general vibes, but lack precision. Microelectrodes, on the other hand, are like those sleek, high-tech earbuds. They can pick up the tiniest whispers from individual neurons, giving us a clear picture of what's going on. Now, before the neurons start their electric chatter, they sit at something called the resting potential. Picture your neuron lounging on the couch, chilling at about -7 millivolts. This is the membrane potential, the baseline electrical charge inside the neuron relative to the outside. To see these electrical potentials in action, we hook up our microelectrodes to an oscilloscope. This nifty device is like the DJ's visualizer at a party, displaying the electrical signals as waves on a screen. Instead of beats, we're watching the spike and dips of neuronal activity. When a neuron gets excited in like a caffeine jolt kind of way, it undergoes depolarization. This means the resting potential moves closer to zero. Imagine our neuron getting off the couch and starting to dance. If the excitement is strong enough to reach a threshold, about 55 millivolts, the neuron fires an action potential. An action potential is the big event, the neuron's way of sending a message down the axon. This is a rapid, dramatic change in membrane potential. The oscilloscope shows a sharp upward spike as the inside of the neuron suddenly becomes positive. This spike travels down the axon like a wave at a sports game, relaying the signal. After the action potential passes, the neuron needs a moment to recover, a bit of downtime, if you will. This hyperpolarization, where the membrane potential briefly dips below the resting potential, is like the neuron taking a breather after all that excitement. The oscilloscope shows this as a downward blip. So there's the sequence. We place our microelectrodes near the neuron, hook them up to an oscilloscope, and watch the neuron go from resting potential, chilling on the couch, to depolarization, getting excited, fires an action potential, dancing wildly, and then hyperpolarizes, catching its breath. Each phase shows up on the oscilloscope as a distinct wave, allowing us to measure and understand these electrical potentials. And there you have it. You got a front row seat into the symphony of neurons in action. Measuring action potentials isn't just about poking neurons with electrodes. It's about visualizing the electrical dance that underpins every thought, movement, and sensation. So next time you think of neurons, imagine them partying in your brain, with microelectrodes as their DJ, the oscilloscope as their light show, and the electrical potentials as the music that makes it all happen. And remember, in the world of neurons, there's never a dull moment. Now, let's chat about diffusion and how temperature makes molecules move like they're in a dance-off. Picture a crowded dance floor. Diffusion is like all those people spreading out to fill every corner of the room. Molecules naturally move from one area where they are packed tight, high concentration, to where there's more space, low concentration. They're just trying to get some more elbow room, if you know what I mean. Now, let's crank up the temperature. Temperature as it's measured in Kelvin, which is the best way to measure temperature. It's like the DJ playing the hottest tracks. The higher the temperature, the faster the music. The more energetic the dancers, aka the molecules, become. They start moving quicker and bumping into each other more often and spread out faster. Kelvin is the scale we use to measure this heat energy. At higher temperatures, molecules get more kinetic energy, making them move faster. It's like turning up the DJ's playlist from a slow waltz to a high-energy dance track. The molecules can't help but move faster and spread out quicker, increasing the rate of diffusion. So the diffusion rate of molecules is directly related to the temperature in Kelvin. More heat means more molecules boogieing, which means a quicker diffusion rate. It's all about keeping the dance floor lively and making sure those molecules are spreading the party vibes evenly. So next time you think of diffusion, imagine those molecules grooving to a beat. And remember, the hotter the beat, the faster the feet. All right, let's dive into the electrifying world of electrostatic pressure, where electrolytes, ions, and other forces come into play. Think of it as the universe's way of keeping charged particles in check. First off, let's meet our main characters, electrolytes and ions. Electrolytes are substances that dissolve in water to form ions. When you dissolve table salt, sodium chloride in water, it splits into sodium and chloride ions. These ions are the VIPs in the electrostatic pressure game. Now imagine a party where opposites really do attract. Positive ions and negative ions are irresistibly drawn to each other, while like charges repel. This force of attraction and repulsion between charged particles is what we call electrostatic pressure. Electrostatic pressure works tirelessly to balance charges. When there's an imbalance, it's like a group of people at the party are trying to get away from someone who won't stop talking about their stamp collection or something. They move around until everyone feels comfortable again. In biological systems, this dance of ions is crucial. Cells, for instance, use electrostatic pressure to maintain their resting membrane potential. Inside the cell, you have a mix of ions like potassium and negatively charged proteins, while outside, there's more sodium and chloride. The cell membrane acts like a bouncer at a party, selectively allowing ions to pass through. The inside of the cell is typically more negative compared to the outside, thanks to the high concentration of negatively charged proteins and potassium ions. The difference in charge across the membrane creates the membrane potential. Electrostatic pressure wants to balance these charges. For example, positively charged sodium ions are attracted to the negative interior of the cell, but they can't just waltz in because the cell membrane is selective about who gets in. Potassium ions, on the other hand, tend to leak out because they're repelled by the positive charge building up or outside. This controlled movement of ions is essential for the cell's function. Enter the sodium-potassium pump, the bouncer that keeps the party under control. This pump actively transports sodium ions out of the cell and potassium ions into the cell, using energy from adenosine triphosphate. It works against the concentration gradient and electrostatic pressure, ensuring the inside of the cell remains negative and the outside remains positive. So let's break it down further. If there's too much sodium inside the cell, electrostatic pressure pushes sodium out to balance the charges. If potassium is too high outside, it pushes it back in. This constant push and pull is what keeps the cell environment stable, ready to respond to stimuli. What's the big picture? Electrostatic pressure is fundamental to many physiological processes. It's behind nerve impulses, muscle contraction, and even the way your heart beats. When a nerve cell gets excited, the sudden rush of sodium ions into the cell and the exit of potassium ions is what generates an action potential, the electrical signal that travels along nerves. In essence, electrostatic pressure is like an unseen force at a party, ensuring everyone is in the right place, balancing attraction and repulsion, and keeping the cellular environment in check. Without it, cells couldn't function properly. Our bodies wouldn't be able to perform the myriad of tasks that keep us alive and kicking. So next time you think about how your body works, remember the ions, the membrane potential, and the relentless balancing act of electrostatic pressure, nature's way of keeping the party going smoothly. Now let's embark on a journey through the microscopic world of the ions, where tiny particles play a big role in keeping our cells alive and kicking. We'll talk about sodium-potassium transporters, organic anions, and the fascinating interplay between sodium, potassium, and chloride in intracellular and extracellular fluids. So grab your imaginary microscope and let's delve in. Imagine your body as a bustling city with cells as its buildings. Each cell has an interior, intracellular fluid, and an exterior, extracellular fluid, separated by a cell membrane, the city's walls, so to speak. Now, let's meet the residents of these neighborhoods. Ions like sodium, potassium, and chloride, alongside with some organic anions that hang out inside the cell. Every city needs order, and in our cellular city, the order is maintained by sodium-potassium transporters, also known as pumps. Think of them as diligent bouncers at the cell membrane nightclub, working tirelessly to keep the party in balance. For every 3 sodium ions they kick out of the cell, they let 2 potassium ions in. This creates a stable yet slightly charged environment inside the cell, essential for maintaining the cell's resting potential. Sodium ions are the social butterflies of the extracellular fluid. They love hanging out outside the cell. Kept in high concentrations by those hardworking sodium-potassium pumps. Meanwhile, potassium ions prefer the cozy intracellular environment where they are in high concentration. When the sodium and potassium move across the cell membrane, they do so either through channels or with the help of those sodium-potassium transporters. This movement is crucial for many cellular functions, like generating nerve impulses and muscle contractions. Chloride ions are the neutralizers. They balance out positive charges of sodium and potassium, maintaining overall electrical neutrality. While sodium and potassium are busy moving in and out of the cell, chloride ions quietly slip through their channels, ensuring everything stays balanced. Inside the cell, you'll find large organic anions, which are negatively charged molecules that can't easily cross the cell membrane. These are like the introverted folk who never leave their homes. They contribute to the negative charge inside the cell, making it a little more electronegative compared to the outside. Diffusion is the natural movement of ions from areas of high concentration to low concentration. It's like people moving from a crowded party room to a quieter one. But our ions also feel the force of electrostatic pressure. The attraction between opposite charges and the repulsion between like charges, this force helps drive ions across the cell membrane, adding another layer of control to the cell's environment. Imagine the cell membrane as a dance floor like we talked about earlier. Sodium ions want to leave the cell, are constantly pumped out by the sodium-potassium transporters, while potassium ions are brought back in. This creates a situation where the outside of the cell becomes more positive due to their higher sodium concentrations, and the inside remains slightly negative because of the potassium and organic anions. This is a delicate dance, but it's essential for cell function. This balance of ions across the cell membrane is not just a microscopic detail. It's vital for life. When you touch something hot, your neurons fire off signals thanks to the rapid movement of sodium and potassium ions. Your muscles contract, allowing you to pull your hand away, all orchestrated by these tiny players. So Next time you think about what keeps your heart beating or your muscles moving, remember the sodium-potassium transporters, the diligent ions, and the forces of diffusion and electrostatic pressure. It's a complex ballet happening at a microscopic level, making sure your body runs smoothly. And just like any great party, it's all about balance, coordination, and a little bit of electrostatic attraction. Let's take another journey into the world of neurons and action potentials. Imagine your neurons as rock stars performing electrifying concerts. To really appreciate the show, we need to understand the role of ion channels, voltage-dependent ion channels, and how the membrane's permeability changes during an action potential. So grab your backstage pass and let's get started. Before the neurons get its groove on, it's chilling backstage at a resting potential of about 70 millivolts. This is the neuron's calm before the storm state, where it's ready but not yet firing. The sodium-potassium pump, our diligent bouncers, have set up a nice environment with more sodium outside the cell and more potassium inside. The scene is set for an epic performance. Ion channels are like VIP entrances at the concert. They allow specific ions to pass through the neuron's membrane, but only when the conditions are right. There are different types of ion channels, but the stars of our show are the voltage-dependent ion channels. These channels open and close in response to the change in the membrane's potential. Think of them as gates that open when the neuron reaches a certain level of excitement. The concert kicks off with a little excitement, a small stimulus that causes the membrane's potential to become less negative, closer to zero. If this stimulus is strong enough to reach the threshold, about negative 55 millivolts, it's showtime. Voltage-dependent sodium channels fly open and the sodium ions rush into the neuron. This rapid influx of positively charged sodium ions causes the membrane potential to shoot up, like a crowd going wild as the band hits the stage. This spike in membrane potential is the action potential, the neuron's big moment. The influx of sodium ions makes the inside of the cell more positive compared to the outside. This positive change propagates along the axon like a wave of excitement spreading through the audience. The action potential travels down the neuron, carrying the electrical signal rapidly. After the peak of the action potential, the neuron needs to calm down. Voltage-dependent sodium channels close, and the voltage-dependent potassium channels open. Potassium ions rush out of the cell, bringing the membrane potential back down. It's like the band playing a slow song to cool down the crowd. This process is called repolarization, and it restores the negative charge inside the neuron. Sometimes the neuron goes a bit overboard with the calm down process, and the membrane potential becomes more negative at the resting potential. This hyperpolarization, akin to the band playing a lullaby after the concert, it ensures that the neurons won't fire another action potential too quickly, giving it a moment to reset. Finally, the sodium-potassium pump gets back to work, restoring the resting potential by pumping sodium out and potassium back in. The neuron is ready for the next big show with its membrane potential back to its chill negative 70 millivolts. During this whole concert, the permeability of the neuron's membrane changes dramatically. Initially, the membrane is more permeable to potassium thanks to those potassium channels. But during the action potential, it becomes much more permeable to sodium as those voltage-dependent sodium channels open. This shift in permeability is what drives the change in membrane potential and allows the action potential to occur. So, the action potential is like a neuron's electrifying concert, complete with opening acts, main events, and cooldowns. Voltage-dependent ion channels are the gates that control the flow of ions, changing the membrane's permeability and driving the neuron's performance. It's a complex but beautifully orchestrated process that allows neurons to communicate and keep the show of life running smoothly. And remember, every time you think, move, or feel, it's because your neurons are rocking out one action potential at a time. All right, let's dive into the electrifying world of action potential conduction, where we'll explore the all or nothing law, rate law, and the flashy phenomenon known as saltatory conduction. Buckle up, because this is going to be a wild ride. First, let's talk about the all or nothing law. This principle is like your friend who either goes all out at a party or doesn't show up at all. When it comes to action potentials, it's all about threshold. If the stimulus is strong enough to hit that magic number of around negative 55 millivolts, the neuron fires an action potential. If it doesn't, well, nothing happens. There's no half-assing it in the neuron world. It's either a full-blown electrical storm or dead silence. Once an action potential kicks off, it needs to travel down the neuron's axon to spread the message. Think of it as a hot piece of gossip that's too juicy not to share. The action potential moves like a wave, with each segment of the axon getting in on the action. Sodium ions rush in, causing depolarization, and then potassium ions rush out to repolarize. It's a bit like a saucy dance with sodium and potassium taking turns on the dance floor. Now, let's get into rate law. This is where the frequency of action potential comes into play. Imagine you're trying to get someone's attention at a bar. You could whisper sweet nothings or shout their name repeatedly. The louder and more frequent your calls, the more likely they'll notice you. Similarly, the stronger the stimulus, the higher the frequency of action potential fired. It's not about the size of the action potential, they're all the same size, but how often they are fired. So size doesn't matter, but frequency does. Kind of like how often you hit the gym rather than how long you stay there. Here's where things get really exciting. Saltatory conduction. Picture your neuron axon as a long winding road. In regular conduction, the action potential would trudge along the entire length, like a boring, slow road trip. But with saltatory conduction, it's more like a high-speed chase scene in a movie, hopping from one point to another. This is thanks to the myelin sheath, a fatty layer that insulates the axons, created by Schwann cells. The myelin sheath isn't continuous. It has gaps called nodes of Ranvier. These nodes are like pit stops on a racetrack. The action potential jumps from one node to the next, skipping the myelinated sections. This leapfrogging significantly speeds up the process, making saltatory conduction of neurons the express lane. Saltatory conduction is like the neuron's version of taking a freeway instead of a scenic route. It makes nerve impulses travel much faster and more efficiently. It is crucial for quick reflexes, sharp thinking, and any activity where speed is of the essence, like dodging a flying beer at a rowdy party. So, the conduction of action potentials is a mix of all or nothing commitment, frequency, and firing at high speeds. The all or nothing law ensures that the neuron doesn't do anything halfway. The rate law determines how often those action potentials fire, and saltatory conduction makes sure they get where they need to quickly. In the end, your neurons are like a crew of party animals, always ready to go out, spreading the electrical love at lightning speed. And just like any good party, it's not about how long you stay, it's about how often you show up and bring the energy. All right. Let's delve into the fascinating world of neuron communication. Imagine neurons as chatty neighbors gossiping over the garden fence, using chemical signals to transmit messages. This conversation happens at the synapses, where one neuron sends a signal to another. So grab your cup of coffee and let's explore how neurons communicate. Synapses are the junctions where neurons connect to pass along their messages. Think of the synapses as cozy cafes where neurons meet up. There are two main parts, the presynaptic membrane on the sending neuron and the postsynaptic membrane on the receiving neuron, with a little tiny gap called the synaptic cleft in between. In this cafe, the presynaptic membrane is like the barista's counter, full of synaptic vesicles, these little tiny containers holding neurotransmitters, the chemical messengers. The postsynaptic membrane is like the customer's table, lined with receptors waiting for their order. The synaptic cleft is the space between the counter and the table where the magic of neurotransmission happens. Synaptic vesicles are like little cups filled with neurotransmitter coffee, When the action potential, the neuron's message, reaches the presynaptic terminal, it's like an order being placed. These vesicles move to release zones at the presynaptic membrane, ready to release their contents. The release zone is the hotspot where synaptic vesicles fuse with the presynaptic membrane to release neurotransmitters into the synaptic cleft. This process is triggered by calcium ions, which enter the presynaptic neuron where the action potential arrives. It's like the barista pouring coffee into the air for a perfectly aimed toss to the customer's cup. Once the neurotransmitters are released into the synaptic cleft, they travel across the narrow gap to reach the postsynaptic membrane. It's a short journey, but crucial for the message to be delivered accurately. Imagine the coffee sailing through the air towards the waiting customer. On the postsynaptic membrane, there are receptors with specific binding sites for neurotransmitters. These receptors are like the customer's hand reaching out to catch the flying coffee cup. When the neurotransmitter binds to these sites, they cause changes in the postsynaptic neuron. The binding of neurotransmitters to receptors generates a postsynaptic potential, or PSPs, which are changes in the electrical charge of the postsynaptic neuron's membrane. There are two types of PSPs, excitatory EPSPs, which make the neuron more likely to fire an action potential, and the inhibitory PSPs, which is IPSP, which makes it less likely. It's like the customer either getting a caffeine buzz, EPSPs, or feeling more relaxed, IPSPs. Many synapses occur on dendritic spines, small protrusions on the neuron's dendrites. These spines increase the surface area for synapses, allowing for more connections and communication points. They're like extra tables at the cafe, making it easier for more neurons to join the conversation. Neurotransmitters act as ligands, chemical keys that fit into specific receptor locks. When a neurotransmitter binds to its receptor, it can open an ion channel or activate a second messenger system, leading to change in the neuron's activity. It's like the coffee's caffeine interacting with the customer's brain to wake them up or to calm them down. Neuron communication is a complex but beautifully orchestrated process. It starts with an action potential arriving at the presynaptic terminal, causing synaptic vesicles to release neurotransmitters into the synaptic cleft. These neurotransmitters bind to receptors on the postsynaptic membrane, generating postsynaptic potentials that can excite or inhibit the receiving neurons. This intricate dance ensures that neurons can effectively communicate, allowing us to think, feel, and move. So next time you enjoy a cup of coffee, remember the intricate cafe-like communication happening in your brain, with neurons chatting away, ensuring you stay awake and engaged in the world around you. Cheers to the marvelous neural network. Now let's delve into the fascinating world of neurotransmitter release and receptor activation. Imagine this as an intricate dance between molecules, orchestrating everything from your thoughts to your movements. So put on your dancing shoes and let's explore the detailed process of how neurotransmitters are released and how they activate receptors, focusing on postsynaptic receptors, neurotransmitter-dependent ion channels, metabotropic receptors, G proteins, and second messengers. The story begins with an action potential traveling down the axon of the presynaptic neuron, a bit like a rock star striding towards the stage. When this electrical impulse reaches the axon terminal, it triggers the opening of voltage-dependent calcium channels in the presynaptic membrane. Calcium ions, always eager to join the party, flood into the neuron. The influx of calcium acts like a signal for synaptic vesicles to move towards the presynaptic membrane. These vesicles are packed with neurotransmitters, the chemical messengers ready to cross the synaptic cleft and convey their messages to the next neuron. The vesicles merge with the membrane in a process called exocytosis, releasing neurotransmitters into the synaptic cleft. It's like the rock star throwing gifts into the crowd. Once released, neurotransmitters diffuse across the synaptic cleft. the tiny gap between the presynaptic and postsynaptic neuron. This journey is brief but crucial. Neurotransmitters reach their target, postsynaptic receptors, without being degraded or taken up by transporters. On the postsynaptic membrane, there are specific receptors waiting for neurotransmitters. These receptors can broadly be classified into two types: ionotropic receptors, neurotransmitted dependent ion channels, or metabotropic receptors. Ionotropic receptors are like bouncers at the club door. The neurotransmitters ligand binds to these receptors. They open ion channels directly, allowing ions to flow into or out of the neuron. This causes an immediate change in the membrane potential of the postsynaptic neuron, leading to either an excitatory postsynaptic potential or an inhibitory postsynaptic potential. For instance, when glutamate, an excitatory neurotransmitter, binds to its ionotropic receptor, it typically opens sodium channels, allowing sodium ions to enter the neuron. This influx of positive ions depolarizes the postsynaptic membrane, making it more likely to fire an action potential. Conversely, GABA, an inhibitory neurotransmitter, binds to its ionotropic receptors. It opens chloride channels, allowing chloride ions to enter. This influx of negative ions hyperpolarizes the membrane, making it less likely to fire. Metabotropic receptors, on the other hand, are like the VIP hosts. When the neurotransmitter binds to these receptors, it doesn't directly open the ion channel. Instead, it sets off a chain of events inside the cell, starting with the activation of G proteins. G proteins are like the messengers who run back and forth between receptor and various cellular targets. When the neurotransmitters bind to the metabotropic receptors, it activates the G proteins, causing it to exchange GDP and GTP. This active G protein then dissociates into two parts, the alpha subunit and the beta gamma dimmer. These subunits can go on to activate or inhibit other proteins and enzymes within the cell. One of the most important targets of the G proteins is the enzyme adenyl-cyclase, which converts ATP to cyclic AMP, a common second messenger. Second messengers like CAMP amplify the signal initiated by the neurotransmitter and can activate a range of intracellular processes. For example, CAMP can activate proteins like kinase A, pKa, which then phosphorylates various proteins, including ion channels. This can lead to change in the cell's ion permeability, gene expression, and metabolic activity. It's like the VIP host calling in more staff to adjust the club's lighting, music, and the bar service to enhance the party's atmosphere. Here's the termination of the signal, the aftermath of the termination of the signal. Once the neurotransmitter has done its job, the signal must be terminated to prevent continuous activation of the postsynaptic neuron. There are several ways this can happen. The first one is reuptake. Transporter proteins on the presynaptic neuron or nearby glial cells reabsorb the neurotransmitter from the synaptic cleft. It's like the club staff cleaning up after the party. Enzymatic degradation. Enzymes in the synaptic cleft break down the neurotransmitters. For instance, acetylcholinesterase breaks down acetylcholine into acetate and choline. Neurotransmitters can simply diffuse away from the synaptic cleft over time. Acetylcholine is the multifunctional neurotransmitter. Acetylcholine is a versatile neurotransmitter that plays a crucial role in both the central and peripheral nervous system. In the central nervous system, acetylcholine is involved in promoting wakefulness, enhanced attention, and facilitating learning and memory. It acts in various brain regions, including the hippocampus, where it helps consolidate new memories. In the peripheral nervous system, acetylcholine is a primary neurotransmitter at the neuromuscular junctions, where it stimulates muscle contraction, and in the autonomic nervous system, where it regulates functions such as heart rate and digestion. Acetylcholinesterase, the enzyme that regulates. Acetylcholinesterase is the enzyme responsible for breaking down acetylcholine in the synaptic cleft. After acetylcholine is released from the presynaptic neuron and binds to its receptors on the postsynaptic membrane, it needs to be quickly removed to prevent continuous stimulation of the postsynaptic neuron or muscle fiber. Acetylcholinesterase accomplishes this by hydrolyzing acetylcholine into acetate and choline. This rapid degradation ensures that the neurotransmitter's action is brief and precisely controlled. The process of acetylcholine breakdown by acetylcholinesterase is a primary example of enzymatic deactivation. This mechanism is essential for terminating the action of a neurotransmitter, thereby resetting the synapse for the next signal. Without enzymatic deactivation, neurotransmitters like acetylcholine would accumulate in the synaptic cleft, leading to prolonged and unregulated activation of receptors. This could result in continuous muscle contraction known as tetany, or persistent stimulation of neurons, which can disrupt normal brain functions. Acetylcholine plays a vital role in muscle movement, cognitive functioning, and autonomic control. Its action is tightly regulated by acetylcholinesterase, which ensures the neurotransmitter is quickly degraded after its release. This enzymatic deactivation is crucial for maintaining precise control over neural signaling, allowing for the fine-tuned communication necessary for complex bodily and cognitive functioning. The communication between neurons isn't just about single signals. It's about integrating multiple signals and adapting over time. The neurons receive inputs from many other neurons throughout thousands of synapses. They must integrate excitatory and inhibitory signals to decide whether to fire an action potential. This complex decision-making process is like a busy switchboard operator managing countless calls simultaneously. Moreover, synaptic communication is dynamic. Synaptic plasticity allows the strength of synapses to change over time based on activity levels. Long-term potentiation, LTP, and long-term depression are mechanisms underlying learning and memory. LTP strengthens synapses, making future neurotransmission more efficient, while LTD weakens them. So, in summary, the release of neurotransmitters and activation of receptors is a sophisticated dance of molecules, crucial for brain functioning. From the arrival of the action potential at the presynaptic terminal to the binding of neurotransmitters to postsynaptic receptors, each step is carefully coordinated. Ionotropic receptors provide quick responses by directly opening ion channels, while metabotropic receptors often offer a slower but more versatile response through G proteins and second messengers. The termination of neurotransmitter signals ensures that neurons can reset and be ready for the next message. This elegant communication system allows our brains to perform an incredible array of tasks, from the simplest reflexes to the most complex thoughts. It's a testament to the beauty and complexity of the nervous system, ensuring that every neuron knows its role and every message gets through, keeping the symphony of the brain playing in perfect harmony. Postsynaptic potentials, or PSPs, are crucial elements in the communication between neurons, determining whether the neuron will fire an action potential. These electrical changes in the postsynaptic neurons' membranes potentials are triggered by the binding of neurotransmitters released from the presynaptic neuron. Let's explore the effects of postsynaptic potentials, distinguishing between excitatory and inhibitory types, and understand their role in neuronal communication and overall brain functioning. Excitatory postsynaptic potentials occur when neurotransmitters bind to receptors that cause an opening of ion channels, allowing positively charged ions, such as sodium, to flow into the postsynaptic neuron. This influx of positive ions depolarizes the membrane, making the inside of the neuron less negative relative to the outside. If this depolarization reaches a certain threshold, it can trigger an action potential, allowing the neuron to send a signal down the axon. The key effects of excitatory postsynaptic potentials. Threshold achievement. EPSPs bring the neuron's membrane potential closer to the threshold, requiring it to fire an action potential. When EPSPs occur in rapid succession or simultaneously at different synapses, they can summate temporal and spatial summation, increasing the likelihood of reaching the action potential threshold. Neural excitability. By making the neurons more likely to fire, EPSPs enhance neural excitability, facilitating the transmission of information across neural circuits. This excitability is crucial for processes like learning, memory, and muscle contractions. Synaptic plasticity. ESPs contribute to synaptic plasticity, the ability of synapses to strengthen or weaken over time. Long-term potentiation, a mechanism underlying learning and memory, involves the strengthening of synapses through repeated stimulation and the subsequent increase in EPSPs. In contrast, inhibitory postsynaptic potentials occur when neurotransmitters bind to receptors that open ion channels, allowing charged ions such as chloride to flow into the neuron, or positively charged ions such as potassium to flow out. This hyperpolarizes the membrane, making the neuron more negative related to the outside and moving the membrane potential further from the threshold for an action potential. Key effects of inhibitory postsynaptic potentials. Action potential prevention. By hyperpolarizing the membranes, IPSPs make it less likely for the neuron to reach the action potential threshold. This inhibitory effect is crucial for regulating neuronal activity and preventing excessive firing that could lead to excitotoxicity. Neural inhibition. IPSPs play a vital role in neural inhibition, ensuring that neural circuits do not become overactive. This inhibition is essential for functions such as motor control, where precise timing and regulation of muscle activity is required. Balancing excitatory inputs. IPSPs balance the excitatory inputs to a neuron, maintaining a proper equilibrium in neural networks. This balance is crucial for cognitive functioning, sensory processing, and emotional regulation. A single neuron typically receives inputs from thousands of synapses, both excitatory and inhibitory. The neuron's membrane potential at any given moment is the result of the summation of all EPSPs and all IPSPs it receives. This integration process, known as neural integration, determines whether the neuron will fire an action potential. Key aspects of neural integration. Temporal summation. Temporal summation occurs when multiple PSPs arrive at the synapse in rapid succession. If EPSPs arrive closely spaced in time, their effect can add up, increasing the likelihood of reaching the threshold for an action potential. Spatial summation happens when PSPs from different synapses on the same neuron occur simultaneously. This combined effect of these PSPs can move the membrane potential closer to or further from the threshold. Threshold dynamics. The overall effect of integrated PSPs on the membrane potential determines whether the neuron reaches a threshold for firing an action potential. This threshold dynamics are essential for the neuron's ability to process complex information and to respond appropriately. So what's the big picture of all of this? The intricate dance between EPSPs and IPSPs through neural integration is fundamental to brain functioning. They allow for precise control of neural activity, enabling the brain to perform a wide range of tasks, from basic reflexes to complex cognitive processes. The key implications of this are learning and memory. The balance of EPSPs and IPSPs is crucial for synaptic plasticity, particularly in learning and memory. The ability to strengthen and weaken synaptic connections through PSPs underlines our capacity to learn and remember. Motor control. In the motor system, the integration of PSPs ensures smooth and coordinated muscle movements. The precise timing and inhibition of neural signals are necessary for activities like walking, speaking, and writing. Emotional regulation. PSPs play a role in regulating emotional responses. The balance between excitatory and inhibitory impulses affects how we process and respond to emotional stimuli. Postsynaptic potentials are fundamental to neuronal communication and brain functioning. EPSPs and IPSPs, through their effect on the membrane potential and neurointegration, determine whether the neuron will fire an action potential. This integration of these potentials allows neurons to process and respond to complex inputs, enabling a wide range of physiological and cognitive functioning. Understanding the effects of PSPs in the neural integration provides insight into the intricate mechanisms of the brain and highlights the delicate balance required for its optimal performance. Now we're going to talk about autoreceptors, which are specialized receptors located on the presynaptic neuron that bind to the neurotransmitters released by that neuron. Think of them as neurons' self-monitoring system. When neurotransmitters bind to these autoreceptors, they typically inhibit further release of the neurotransmitters or reduce the synthesis of neurotransmitters. This feedback mechanism helps maintain neurotransmitter levels within the optimal ranges, preventing excessive stimulation and ensuring balanced neurocommunication. So what are some of the different types of synapses? Axioaxonic synapses occur when the axon of one neuron forms a synapse with the axon of another neuron. These synapses play a crucial role in modulating the amount of neurotransmitters released by the postsynaptic axon. Two key processes facilitated by axioaxonic synapses are presynaptic inhibition and presynaptic facilitation. Presynaptic inhibition is, in this process, the action of the presynaptic neuron reduces neurotransmitter release from the postsynaptic neuron. This can occur through mechanisms like reducing calcium ion influx, which is essential for neurotransmitter vesicle fusion and release. It acts as a braking system, fine-tuning neural signals to prevent overstimulation. Presynaptic facilitation enhances neurotransmitter release from the postsynaptic neuron by increasing calcium ion influx. More neurotransmitter vesicles fuse with the membrane, amplifying the signal. This mechanism acts like an accelerator, boosting neural communication when needed. Gap junctions are unique structures that allow direct electrical communication between adjacent neurons. They are formed by connexin proteins that create channels linking the cytoplasm of two neurons. Through these channels, ions and small molecules can pass directly from one neuron to another, enabling rapid and synchronized activity. Gap junctions are crucial for processes that require fast, coordinated responses, such as certain reflex and rhythmic activities in the brain and heart. Understanding the role of autoreceptors, axioaxonic synapses, and gap junctions provide a deeper insight into the complex regulatory mechanisms of neural communication. These systems ensure that neural signals are precisely controlled, maintaining the delicate balance necessary for effective brain functioning and overall neural health. Let's venture beyond the traditional synaptic conversation and explore the world of non-synaptic chemical communication. Here, we'll encounter neuromodulators, peptides, hormones, steroids, and their favorite pen pals, target cells. It's like a grand opera where everyone has a role, but not everyone is center stage. Think of neuromodulators as social media influencers of the nervous system. They don't directly cause action potentials, but tweak the mood of the neurons, making them more or less likely to fire. Neuromodulators are released into the extracellular fluid, diffusing over large areas and affecting multiple neurons. It's like adding a general ambiance to a room rather than whispering secrets. Peptides are short chains of amino acids that function as both neurotransmitters and hormones. They're the multitaskers, the busy bees. For instance, endorphins, the body's natural painkillers, are peptides that can act locally with the brain to modulate pain receptors. Peptides are like the versatile actors who can perform on stage, in synaptic communication, and behind the scenes in non-synaptic roles. Hormones are the marathon runners of chemical communication. Released by endocrine glands into the bloodstream, they travel long distances to reach their target cells. Imagine sending a letter by snail mail rather than a quick text. For example, adrenaline produced by the adrenal glands boosts your fight or flight response, affecting various organs throughout the body. Hormones ensure that messages get delivered far and wide, coordinating complex body functions. Steroids are a type of hormone with a bit more oomph. Derived from cholesterol, these lipophilic or fat-loving molecules easily slip through cell membranes to reach their receptors inside target cells. They're like VIPs with all access passes. Cortisol, a steroid hormone, helps regulate metabolism and immune responses. Steroids don't just knock on the cell door, they waltz right in and start making changes from within. Target cells are the ones tuned into specific frequencies of these chemical signals. They have receptors that match the molecular shape of the neuromodulators, peptides, hormones, and steroids. It's like having the right app to read the QR code. When these molecules bind to their receptors, they trigger various cellular responses, from gene expression changes to altering the cell's metabolic activity. Non-synaptic chemical communication is like a grand symphony where each molecule plays a crucial role, creating a harmonious balance within the body. Neuromodulators set the mood, peptides juggle multiple roles, hormones send long-distance messages, and steroids make powerful internal changes, all while target cells act as the attentive audience. So next time you think about how your body maintains its delicate balance, imagine this complex dance of molecules. Each has its own unique role, ensuring everything runs smoothly, from your stress responses to your morning jog. It's a beautifully orchestrated performance that keeps you thriving day in and day out. Let's bring this brainy journey into a close with a few thoughts on why understanding these tiny molecular dances is so important for our health and mental prowess. The brain's secret sauce. Knowing how neurotransmitters, neuromodulators, peptides, hormones, and steroids works gives us the secret recipe for the brain's secret sauce. It's like having keys to the kingdom, or at least to the brain's control room. With this knowledge, we can better appreciate how our thoughts, emotions, and actions are finely tuned by these chemical messengers. Mental health mastery. Understanding these processes is crucial for mental health. Conditions like depression, anxiety, and even schizophrenia are often linked to imbalances in neurotransmitters and hormones. It's like having a symphony where the violinist is playing too fast and the drummer is out of sync. By knowing the role of these chemicals, we can develop better treatments and therapies, helping to restore harmony in the brain. Boosting cognitive power. Do you want a powerful mind? This knowledge is your supercharger. By understanding how synaptic and non-synaptic communication works, you can make lifestyle choices that boost your cognitive functioning. For instance, regular exercise increases the release of endorphins and growth factors, enhancing mood and brain health. Eating a balanced diet ensures your brain has the right building blocks for neurotransmitters. Think of it as giving your brain the premium fuel it needs to run like a high-performance engine. Longevity and well-being. Let's not forget the role of hormones and peptides in overall health. They regulate everything from metabolism to stress response. Understanding their functions can help us age gracefully, stay resilient in the face of stress, and maintain a healthy weight. It's like having a personal health coach inside your body, guiding you towards optimal wellness. So here's some final thoughts. Next time you're marveling at your ability to solve complex problems, remember the intricate dance of neurotransmitters, hormones, and all their buddies. They're the unsung heroes working tirelessly behind the scenes with little knowledge and a lot of appreciation. We can harness their power to enhance our health and develop a truly powerful mind. After all, understanding your brain's chemistry is the first step in mastering your own destiny. Cheers to your brain, the ultimate maestro of your life's symphony.